So now let's move on to the second part of this lecture, seed banking. To produce a lot of seedlings, we need a lot of seeds. So make sure that at least once a month you have seed collection trip. In this picture is an example of one seed collection trip that we get the dry fruit. And collecting seed should be done when the seeds are ready to disperse. And how we gonna notice when the seeds are ready? We can see from the changing of the color of the fruit. For example, in this picture, these are dry fruit. So immature fruit will maybe green, and then once it change into brown and look dry, it's probably ready for collection. And also you can observe the trees a little bit more, and if you see that birds or squirrels are interact with the tree, they may come and eat the ripe fruit of the tree. That is also a good sign that the fruits are ready to be collected. We want to make sure that we do not collect all the seeds from only one tree, because we want to increase the genetic diversity of our seed collection. So make sure that at least 10 individual trees provide the seeds into seedling production. And if possible, try to collect the seeds from the tree. But sometimes if it unavailable on the tree, it's already fell down to the ground, it's also okay to collect seed from the ground, but we prefer the seed that are collected directly from the tree, because they are not exposed to fungi and or some bacteria that will make the seed have low quality. And after we collect seed, we can use the flotation technique to identify the good quality seeds. And by flotation technique is that after you get the seeds, you put them in the water, and the seed that floats is not good. And we should keep the seed that are sink into the bottom of the bucket. In this picture is the practice of using the rope with the heavy weight, and we will swing the rope and try to get the rope onto the branch, and then we shake the branch in order to get the seeds when the fruit are dropped. So here is our staff teaching one of the workshop participants how to use this technique for seed collection. One thing that we can do once we have a lot of seeds and we start to accumulate a lot of seeds is to put the seeds in the seed bank. And why seed bank is important and useful? First thing is to keep the seed that we have a lot, right? And then we can distribute those seeds into a different area and exchange the seed with a different project. And also we keep the seed just in case that maybe the next year the tree is not producing a lot of seeds. We can still use the seed in our seed bank to produce seedlings. And also lots of information about seeds are not available. Sometimes you keep those seeds for future research study that you can do with seed. And those research can help you in terms of nursery management and also seed bank management as well. And more than that, if those species are vulnerable for extinction, keeping their seed in the seed bank can help to secure. If those species extinct in the wild, we will still have the seed material for propagating those species and maybe reintroduce into the habitat. And for seed banks can be in a different size. It can be a very big seed bank like the one at the Millennium Seed Bank at Waker's Place. And also it can be a small one. For example, in the Dorsotame Nature Center, we have a small local seed bank. And our seed bank right now consists of 3 freezers. And this is a picture of those 3 freezers that we use. And for seed banking itself, it involves seed collection, identification of the species, seed cleaning and pre-processing, and storing those seeds in the freezers. Moreover, the seed that we keep in the refrigerator from time to time, we take it out to check if the seed still viable. We have to keep in mind that not all the seeds from every tree species can be stored in the seed banking condition. And the condition in the seed storage in the seed bank, as you can see, I show you that we keep the seed in the freezer, and the temperature that we use is minus 20 degrees Celsius. And also, in order to put the seed in the freezer, the seed should be dry as well, because if it's very moist, the ice crystal in the seed can damage the seed. So before we can even put the seed in seed bank, we should know what we call seed behavior, seed storage behavior of each species. So we should start identifying the seed storage behavior of the tree species that you work with. And in general, there are 3 types of seed storage behavior: orthodox seeds, recalcitrant seeds, and intermediate seeds. The first type, the orthodox seeds. Orthodox seeds can be kept in low temperature at minus 20 degrees Celsius, and it can keep in a dry condition. These are the seeds example of the orthodox seed. It's the Phyllanthus emblica. So for orthodox seeds, freezing and drying will not kill the seeds. The second type of seeds is recalcitrant seed. For recalcitrant seed, drying and low temperature kill the seeds. So the recalcitrant seed cannot be kept in seed banks. And here is the example of recalcitrant seed. And the last type of seed storage behavior is intermediate seed. For intermediate seed, it's in between orthodox and recalcitrant. Intermediate seed can be dried to lower moisture content. However, they may not be able to keep at lower temperature. And this is the example of the intermediate seed. It's Magnolia bellonii. In order to identify seed storage behavior, we need experiments. Here's the example of the experiment. And for this experiment, we use wet storage and dry storage. Then we compare the seed viability between the 2 treatments. The left-hand side box is a drying experiment. We put drying beads in white color and the seeds together in the box. And the right-hand side is the wet storage. And the wet storage meaning that we keep the seed with the moist filter paper. And then when we leave the seed dry, and then after that we take the seed out and try to germinate them. And then we compare the germination between the 2 treatments. Once we know the seed behavior, seed storage behavior of each species, we can plan of what species we can keep and what species we cannot keep in the seed bank. For the species that we cannot keep in the seed bank, it's good to just germinate them and keep them as a seedling. But for the species that we can keep in the seed bank, after we collect the seed, we will have to do some pre-processing and cleaning before we can store them. So here is the example. If the species have fleshy fruit, which means that it has a lot of flesh, it's pretty wet, and it has some meat of the fruit around the seed, you should first remove the seed out from the fruit first and then clean it with water. And that is the example on the left hand side here. And the technique for remove the seeds will be different from species to species according to the type of the fruit they have. And for dry fruit that may be in the seed pod, they may have wings, so then we remove the seed from the seed pods. For example, in this picture on the lower left hand side. And then after we remove the seed, we clean the seed, we should dry the seed to low moisture content before we put in the seed bank. And on the right hand side here is easy equipment that can help you dry the seed. We can put the drying beads or the charcoal in the bottom, and then we have in here we have the wire nest that we make into the cylinder shape that allow us to hang the bag of the seed inside and keep them dry gradually. And for storing seeds in the seed bank, aka in the freezer in our local seed bank, we use the temperature at minus 20 degrees Celsius, about plus or minus 3 degrees Celsius. And the moisture content of the seed should be not more than 15% equilibrium relative humidity. This is the example of our freezer. You see that there are two types of containers here, the glass jar and the foil and plastic bag. The good thing for using bag is to, when you have limit space, the bag can stack them together easily and save the space. And this on the left hand side is the example of the bag of the seeds that we store, and we put the name of the species, and if you have S number and batch number, make sure to include them too. When we bank the seed, we should make sure that the seed that we bank is viable, is still good. So we have one easy pre-screening for the viability of the seed by cut test. And cutting test, you just cut the seed and try to see inside if the seed contains embryo. So this is the example of the seed that we cut, and then you see that, oh yeah, this seed is filled, it has embryo in there. You can see the cotyledon and the endosperm in there. So that's okay, that's a good quality seed. Or if you have a pyrene, in one pyrene may have lots of embryo, lots of compartments, for example in here on the right hand side here. And if you cut and then you see that the seeds are filled and have embryo, that means that at least it's possible that the seed will be viable. However, the cut test may not tell you if the seed will be able to germinate or not, but at least you get the sense that, oh, this is a good quality seed. And other thing that we can test, like I say, seeing that the seed is filled is not guarantee germination. So another thing that we can do is we germinate the seed in the seed germination tray, the black one like this, and then we monitor the germination of the seed. So here we come to the end. Today about seed banking, I only talk about it briefly, and I introduce you to the different type of seed storage behavior, and also I introduce you to the brief process from seed collection to seed storage. And thank you for your attention for this lecture, and I hope to see you later. Bye bye.